Excellent. So thank you so much, Dr. Cohen. That was excellent. It's so wonderful you see how closely we all work together, surgical oncologists, medical oncologists, and even our radiation oncologists and other. So now we're going to transition a little from kind of what is standard, although a lot of what we heard from Dr. Meridian, that was really the neoadjuvant approaches, which are rapidly emerging and still we're doing a lot of work in, to a bit more what is coming. And so I'm thrilled now to introduce Dr. Patrick Ott, a close colleague of mine in medical oncology at Dana-Farber here, whose interest is in all sorts of things, including novel therapies for melanoma, but in particular in vaccine therapy and in neoantigen vaccines for melanoma. And so I'm going to hand it over to him to tell you all about that.
Thank you very much. Thanks for the introduction. It was really exciting to be here this morning. I hope this actually works being this far away from the microphone, right? Do you get feedback from people can actually hear? Okay, sounds good. All right, so I wanted to spend a couple of minutes on this slide here because I really wanted to convey what our objective is when we give vaccines for cancer. And Dr. Moraini really gave a great introduction to the T cell already, right? It's also been called that our T cells in our body have also been called the T cell army. But when we do immunotherapy, including actually all as we call it, the checkpoint inhibitors, which is really the PD-1 inhibition, the CTLA-4 inhibition, and the LAG-3 inhibition, mainly, which was also already introduced this morning. But what we really do with these therapies, we want to increase the frequencies of the T cells. So we want to get more of these T cells into the tumor. It's also been called like changing or converting a cold tumor into a hot tumor, right? So we basically want to get as many as possible of these cells in there. And why do we want to do that? What's sort of attractive about T cells? Well, they have memory function, meaning they can actually learn how to do their function over a long time. They can actually learn this and then use it like years later. That's a little bit, we'll get to the COVID vaccine analogy in a little bit, but basically that's what vaccines are all about, right? So you basically get a vaccine and then you're protected years later. The other feature really with the T cells is like how do they work and how do they function? And so there's two different systems in our immune response. There's like what we call the innate immune response, and there's called the adaptive immune response. And the T cells are part of the adaptive immune response, which means that they actually recognize specific antigens. And so in the COVID world, right? So we've all lived through 3 years of like, it feels forever, but it's also like been a lot of like everybody going through like development of vaccine, right? So you could on many different levels, but you could certainly read in the New York Times about like vaccine development, how the different factors. A lot of talk has been about the spike protein, right? So which is basically the antigen of the virus, right? The same thing applies to cancer in a way that we actually want to steer these T cells against antigens, right? But the antigens in cancer are much more complex. Meaning that we don't actually always really know what the antigens are in these cancer cells. And the cancer vaccine field has been around for decades. And one of the major challenges really has been to know what are the antigens that we should actually vaccinate against. And then just the third point on this slide is like, well, most of the vaccines in the infectious disease world, if you will, which is the majority of vaccines, it's typically prophylactic. And you basically vaccinate and then like sort of the virus hits the body, maybe weeks, months, or years later. Versus in cancer, the target or sort of the cancer cell is typically already around. And that could be like, it could be a patient with metastatic cancer, but it could also be like all the settings that we talked about where there is a melanoma that gets surgically resected and we worried about the recurrence, but what it basically comes down to is this micrometastatic disease. But the bottom line is that the cancer cells are already there when you're vaccinating. And that's like a challenge, as you can imagine. And then maybe the fourth point is like, because there's so many antigens, what we think is like, we probably shouldn't vaccinate against just one antigen, but against several. And that goes at that's like we call it clonal heterogeneity. there's actually tumor and cancer evolution, meaning like the cancer cells change over time, the composition changes. And so how do you like do that? How do you account for that therapeutically? We think is like, well, it's not going to be enough to just pick one antigen and then hope that that's going to do the job. And it's actually illustrated with different colors in the picture. And then just, I sort of made the point already, but it illustrates it here. So what is the difference between infectious disease and cancer? Is like on the left side, you see there's this, you actually see this? Oh yeah. So basically when that COVID virus, you get infected with it, the immune system has never seen it. So it's like foreign, and it's by definition immunogenic because it's something that doesn't really belong to the body, right? It's not really something that the immune system has sort of learned not to recognize. Versus for cancer, it's sort of in between. So cancer cells are, not exactly normal body cells, but they have arisen from normal body cells. And they're therefore, our job is to actually figure out what can we actually vaccinate against that's not what the immune system has not already learned that it shouldn't respond against. And then also it's like a long process. It's not something where the virus comes from one day to the next. It's something that those cancer cells we think have sometimes evolved over years, and that the cancer actually has learned to evade the immune response. Okay, how do we vaccinate? There's a lot of talk about RNA, right? So everybody knows about like RNA is a great, what we call a vaccine platform. But actually before COVID, that wasn't the case at all. It was actually that our RNA has never been used as a vaccine before. We actually used, the reason why we call platform is basically we have to find a way to deliver that antigen, and you can do that in different ways. So you can use like peptides, you can use proteins, you can actually use DNA, because it can cause viral vectors. And the whole development history of COVID vaccine also illustrated that too. So different companies used those different platforms. There's different ways to deliver those vaccines. So it can actually, some vaccines are injected, as you know, in the muscle. They can be injected in the skin. Some of the vaccines are actually given IV, but you can also do different intervals. So you can give, again, like the analogy with the COVID. So you get like a prime, so you get your first COVID shot, and then like a couple of weeks later, you get the next one. Now we talk all about boosters, but there's a lot of empiricism to it. So we don't really know whether it's actually the right strategy. And you can imagine, because of the cancer vaccine are so much more complex, there's even more uncertainty, like what is actually the best dose. And then the third point on this slide here is like, probably we actually have to give the vaccines in combination, for example, with checkpoint inhibitors or other agents that can sort of additionally stimulate the immune response. And then now we're getting to personalization, right? And I just wanted to make a comment. So the word neoantigen has already been introduced, and that's important because neoantigens are essentially the proteins that are encoded by mutations, right? So in all tumors have mutations. Melanoma is actually one of the most highly mutated tumors, if not the most highly mutated tumors. So typically in a melanoma, when we find the mutations by sequencing, we can find hundreds, if not thousands of mutations. But there's also a spectrum. So some melanomas have fewer and some have more mutations. But the reason why neoantigens are important is because because we can actually now identify them with sequencing, which really wasn't possible even like 15 years ago, right? So there was simply not the technology to actually sequence a tumor, find out about the mutation for a patient. It would cost like millions of dollars. It would take like months, if not years. But the ability to actually do that on a sort of timely basis and actually also in a reasonable price, so it allowed us to use the neoantigens for vaccines. We know that the neoantigens are actually important for those treatments that are not vaccines, right? The immune checkpoint inhibitors work better in tumors that have more neoantigens. And then the other part is like the personalization, right? So why is this like a personal approach? Well, this is actually illustrated here. Most mutations are what we call private. So there is like, we know about, and they have already been discussed too, like there's this BRAF mutation, right? Half of patients with melanoma have a BRAF mutation. But as I mentioned, there's like hundreds of thousands, which are actually the ones that are that we should vaccinate against, right? So it turns out like, well, ideally you would want to vaccinate against BRAF, right? Because you know it, 50% of the patient have it, why not just doing a BRAF vaccine? Well, the reason for that is, number one, it's only half the patient. Number two, not every patient's immune system can actually even recognize their own BRAF mutation when they have it because of this diversity of HLA. So when you think about like transplants and stuff, so you know that there has to be, that each patient's immune system is different. That's why you have to match donors. And that diversity is reflected in this. So let's say you have like 50% of patients have a BRAF mutation, probably only like 20% of them actually have the right HLA to do a vaccine. So here's where we then get at the personalization. Because we have all these other mutations, we can target hundreds, potentially hundreds of mutations, but they're different in each individual tumor. And so you cannot just pick a couple and then design a vaccine, although that has been tried. But if you want to really make this a treatment that's a therapeutic for all melanoma patients, ideally, you have to personalize. And I just one plug sort of the word personalization. I feel like that's in some sense a little bit overused and there's like some maybe even confusion, right? So you can tailor treatments depending on what we have. So BRAF is actually the best example. Half of melanoma patients have a BRAF, as I mentioned. So we're not only going to give those target therapies those half patients, right? In a way that's personalized. You find out the mutation and give the drug only to half the patients. But of course, like the other 50% that don't have the BRAF mutation, they kind of they don't have a therapy, right? Versus like in my sense, like a true personalized therapy is actually where you custom make the vaccine, right? So basically, or you custom make the therapy, basically account for the differences in the tumors. And then you design a vaccine for each individual patient. And so that is, it's just, I think the advantages are that you don't, it's not stratified. You don't just select patients out based on how their tumor, and then there's sort of the people, the patients who are lucky enough to have that right, like composition of genes or whatever, versus you say like, well, let's make a vaccine. And, you know, we know that we can make a vaccine. We just have to like custom design it. But of course, that's challenging because it takes, of course, money, time, and like there's a lot of complexity to do this customized vaccine. As I've already been mentioned, how do we, and I mentioned just a couple of minutes ago, how do we know what the mutations are? You actually need the tumor, you need a blood sample, you sequence it, and then you call the mutations. That's actually, when you sequence, you have like all these like different, a ton of information that you actually have to make sense out of it. And this is, not easy. It's becoming easier as we have like more computational analysis. But I will say, even with the tools that we have now, it's actually more complicated than most people realize. And then because of this, the HLA that I also mentioned, you have to like, and not only you have to call the mutations, but you also have to really pick which you think are the ones. So out of those hundreds of thousands of mutations in the tumor, you cannot vaccinate against 1,000 mutations. You pick 20 or so, and then you find out like which are the best 20, which are the ones do you think actually the immune system will recognize. And that's basically reflected here, just like pipeline that we use where you have the tissue first, you do all this like analysis, computational analysis, and sequencing, and then you end up with like the right mutations to vaccinate against. And then you make your vaccine. Typically it takes like 3 months, although the timelines are definitely getting quicker. So when we first started this here 10 years ago in melanoma, we used peptide. Our first study was in high-risk melanoma patients, so those same patients that were already essentially had a lymph node metastasis, it was resected, they were free of melanoma, they received the vaccines. I'm not going to go into like too much detail what the vaccines were like, only to say that we actually saw really nice immune responses in all of the patients against many of the antigens that we were vaccinated against. And that was like quite remarkable at the time because like despite, as I mentioned, decades of vaccine design and development, they really hadn't this kind of consistency and robustness of responses that hadn't been seen before. And we actually, in the study really only had 6 patients, but actually 2 out of those 6 patients had metastatic disease, so they had lung metastases. They had, their lung metastasis actually came back while we were vaccinated, sort of illustrating the point that I made earlier, that the vaccine alone may actually not be enough, but at a certain point, it may take more time for the vaccine to work. And so you need something that's not vaccine to treat for a metastatic patient, but they both then got pembrolizumab, and both of them actually had complete response, meaning like regression of all tumors that's maintained till this day, that can happen with PD-1 inhibition alone, but I would say it's at least unusual to have two patients and it happens for both of them. So it's suggesting that because they got the vaccines before the pembrolizumab, they may actually have had sort of this immune response that I was talking about, where you basically get these T cells ready to and have like higher numbers, and then you use the drug that actually then releases those breaks, and that's sort of the ideal combination. We then actually followed these 6 patients longer, and after an average of 3.5 to 4 years, they were all still alive. A few of them actually had recurrences. But the most important thing I would have shown on this slide, so we basically got more blood years later, and then we're able to test the immune responses against the vaccines again after an average of 2.5 to 3 years. And lo and behold, the majority of the immune responses that we saw after like just a few months were maintained after years, which is really, that's sort of exciting because it really shows you that these memory function of the T cells is something that you can induce with the vaccine. And then we did some fancy analysis, but it's also a little bit of a plot, like when we collect tumors and we collect blood, we actually want to learn from this. And there's lots of tools that we have now, some of them called single cell analysis, where we can actually look at single cells, we can look at function of these cells. And with a vaccine, we can actually look at T cells that are specific, that we know are specific for the vaccines, and we can look at their gene panels. And what we showed in that long-term study is actually that those T cells exhibit a memory phenotype. And sort of the function of these T cells was exactly the way we would expect it to be. And then we also looked at, I mentioned like all these different specificities and the different antigens. That is actually taken care of by the T cells because they have what's called a T cell receptor, and they are extremely specific. And what we think that what's happening after vaccination is like there's like this diversification, meaning like that T cell repertoire gets broader and sort of encompasses more specificities. And that's exactly what we saw happening after a couple of years when we looked at, on a single cell level, these T cell receptors of vaccine specific T cells. And so just to point you like this was actually novel enough, and it was recognized that vaccines could work for cancer, and it was considered a pioneering study because it was really like the first time that these new antibodies, these new targets were used, and nature sort of recognized as one of the milestones in vaccine, which I thought is kind of nice because when you see it actually onto like the 1700s and sort of have it stamped as like one of those milestones was kind of nice. And so this was really the first study. There was an effort to broaden this concept, and actually the team here at Dana-Farber founded a company called Immuno-Therapeutics to put this on a larger scale. And so a study was designed where, what I mentioned before, basically go into metastatic disease, and you combine with PD-1 inhibition. I'm just going to show a couple of data points from that trial. So there was like a much larger study that had like 60 patients. The majority of them were melanoma. They received nivolumab while the vaccines were made. They were then vaccinated, and then they continued nivolumab. And again, so those were metastatic patients. And so what we see here is like waterfall plots. I don't think we have seen any this morning, but basically that's a way for us to like look at tumor responses. A bar that goes down is good because it means that the tumor regresses. And what we saw in the melanoma cohort, this is individual patients. There were a couple that had like progression, so the tumors became larger, but the majority actually had regression. And then the difference between these like dark and more brighter bars is like after the PD-1 inhibition, there was already response, and some of the patients actually then had deepening of these responses and further regression after the vaccine. And so this is actually this observation of the PD-1 inhibition, which is the standard of care, doing something, is actually reflected here, where we had patients that had tumor biopsies before the PD-1 inhibition, before the vaccine, and after the vaccine. There was actually 5 patients who had like great responses under PD-1. So that's like obviously what we want to see when we just do standard of care. We have seen already slides. It's essentially you treat, you look at the tumor, and you see, oh, the tumor regressed completely. But there was actually 9 patients who didn't have that. So basically they got normal treatment. They had biopsies, and then the pathologist looked at these biopsies of the tumor, and the tumor had not regressed. But then actually after the vaccine, they had regressed. So basically prior to the vaccine, this year, and then after the vaccine, the tumors had all regressed. And actually patients who had this regression did better than patients who did not. So sort of suggesting that the vaccine had an effect. I'm actually gonna probably skip over this here. It's just like a phenomenon that we call epitope spreading, where we look at whether the immune response actually goes beyond the vaccine. So you're vaccinated, and the immune response actually starts recognizing even antigens that they were not vaccinated against, which could suggest that tumors, sort of the cells actually got killed and exposed more antigens, and that primed more immune responses, which we sort of like to see in immunology, because like that could suggest that there's actually a specific response or a specific killing of tumor cells. So you've seen already this data, so I'm not going to belabor it, but why is there so much excitement? Why is it in the lay press? Like, why is this like a big deal? Well, because it's really the first, this study where 150 patients with melanoma, with stage 3 melanoma, were treated. 100 of them got the standard of care, Keytruda or pembrolizumab, along with an RNA vaccine. The study was done by Moderna. And 50 of the patients only got the standard of care. And so it's called a 2-to-1 randomization. I would say it's like a relatively small study. It's like a phase 2 study that can show us like statistically significant differences, but it's not typically thought to be a study that tells us that it actually works. And so it actually works sort of when it works reliably enough that it should be FDA approved. But we saw a difference, and something like this has never been seen with a cancer vaccine before. That's why there's all this excitement about it. How much time do we have?
Not much. You're on.
All right, so I'm just gonna show one slide here. So we have we actually want to make these vaccines better. We have more trials now. We combine them with more things at Dana-Farber, for example, drugs like montanide and ipilimumab that we inject right next to the vaccine. And basically, just a plug for what we do when we do this tumor biopsies and the leukapheresis is we do a single cell analysis. And I think we were quite excited to have a patient where we followed the tumors over time while they were vaccinated. We saw tumor regression, but we also saw when we looked at these T cell receptors that there was new T cells that were induced after the vaccine on a single cell level. So that's it just tells you a little bit we can do fancy analysis, but they can actually be super informative to help us actually figuring out well, we vaccinate against something, is it actually reactive, and what are the T cell receptors doing? And so that really helps us developing novel or the next iteration or generation of the vaccines. And so there's still quite a few challenges. So we don't really have the best way to really pick the right neoantigens. It's we have pipelines that we think are pretty good, but there's a lot of room to make them better. We can certainly increase the manufacturing capacities, and we can sort of shrink that time frame. And then ideally, we really want to make these vaccines even stronger than they are, meaning we want to what's called prime the T cells better than we currently can do. And we probably also have to combine, as I mentioned, with drugs that can counteract what's going on in the tumor itself. And then we can work on even new antigens. This is just to say that it's the work, you think it's 6 patients on a trial, but there's actually the team that's developing this, at least in the very beginning, there was about 30 to 40 people involved. So thank you.